All right, should we kick things off? All right, welcome everyone. I'm Alex Sabo, the CEO of Millennium Science, and I am absolutely delighted to welcome you all to the webinar, the introductory webinar for the 2025 Millennium Science 10x Genomics Fellowship launch. This is an activity that we've been carrying out now for, this is the sixth iteration of the Millennium 10x Fellowship. And we're absolutely delighted to see that there's once again so much interest in this fantastic program. Just like to tell you that during the 6 years that we've been running this program, um, this activity, it's arguably been the most successful technology adoption program that Millennium Science has ever conducted. Over the course of this program, there have literally been dozens of fellows who've used this essentially as a springboard to get into the single cell and spatial biology field, to secure grants, to publish papers, and in many cases, quite impressively, to proceed from being absolute novices in the field of single cell or spatial biology to becoming prominent speakers, prominent thought leaders in the field here within Australia and New Zealand. And I should say that I'm especially proud of this activity because it's been viewed as so successful that it's been copied actually throughout the world. The first fellowship was set up here in Australia and New Zealand, and similar programs have now been adopted throughout the world, including in the United States, the home of 10x Genomics. So this has been a very valuable program. Um, and I think you'll through the talks that my colleagues Marta, Jeff, Kat, and our honored guests Jennifer and Dimitri will share with you, you'll learn about why that is. But it has to do with the support that we offer in introducing scientists to single cell and spatial biology, and more importantly, the community, the self-supporting community around this important technology field. So I'm going to hand it over to Marta to tell you more about the details of this exciting program. But I just want to say that as you're listening to Marta and my other colleagues here, I hope very much that this piques your interest for how you can apply this groundbreaking technology in your own research and propel your career. Thank you very much for joining us. And on to Marta. Go ahead.
Thank you, Alex. I, as usual, always have some issues with sharing my screen. I hope now it looks okay. So welcome to today's information session about Millennium Science 10x Genomics Single Cell Fellowship 2025. So on the agenda for today, I will be briefly talking about what the fellowship is all about. Then Jeff will give practical introduction to single cell sequencing, followed by Kat, from 10x Genomics, that will talk about the importance of sample preparation. And then we will have discussion with Jennifer Bauer and Dmitry Shek, our fellows from previous years, about how the fellowship looked for them and what advice they might give you. And we will also have time for questions and answers. So what the fellowship is all about. Fellowship, as Alex mentioned, was launched quite a long time ago, but it's, for me as a fellowship coordinator, gives sense of community to the fellows which are accepted to the program. So Millennium Science and 10x Genomics work closely together, so we almost form like a family. And we invite you with this fellowship, we invite you to become part of that family. And that means that it, of course, have several benefits, but we are, all the fellows are always very close to our hearts, and they become friends. So what are the benefits, actually hard benefits of the fellowship? So we'll offer personal mentoring session and experimental design help from 10x Genomics Science and Technology Advisor, which is Jeff, and field application scientist team, which can be Kat or Jerry. What else? The personal care of the fellowship program coordinator, which is myself, Marta Gabrielska, and you can, what it means that you can contact me with any questions, any problems. If you lose the contact details to anyone from the team, or you have a question, you can just do it. We will receive discounted 10x Genomics consumables as part of the fellowship. If you would like to learn how to run Chromium instrument, there is a possibility of receiving a demo Chromium instrument placement. This is subject to availability, but it opens the door to really try the technology hands-on if you don't have the equipment in your institute. Upon the completion of the fellowship, you will receive a certificate that you can then use in your CV or on social media. You will also receive speaking invitations every time we have once a year, we have a fellowship forum event or 10x Genomics user group meetings. You will also be basically member of the 10x Genomics Millennium Science Fellows Network, which means you receive special information, special promotions, and you're basically a very important person to us, and we will treat you as such. And of course, there will also be some fellowship exclusive merchandise. So the application, which is open now, what does it entail? We ask you to provide a project title, 50 words maximum, abstract, no really lengthy outline of what you're planning to do. So it's not like NHMRC or ARC applications that you spent 3 months writing. Also, please attach 1-page CV and a letter of support from your lab head. So conditions of this fellowship: no previous personal experience with 10x Genomics single-cell technology. We especially encourage graduate students, early to mid-career researchers. Research assistants are also eligible. Just this, what we want to do with the fellowship is to enable really young researchers to have additional step in their career ladders. And we ask you to, that experiments will be carried out by the applicant and not by someone else. So what happens next? Please apply for the fellowship by the end of this month. Selected individuals will be contacted via email. Onboarding meeting will be scheduled, and that's where the fellowship really starts. There will be multiple points of contact during that process to ensure that you're successful in your project, and that ends up with us sending a survey and a certificate at the very, very end. So not all those who wander are lost. Some of us just want to do single cell sequencing, but just to not get too lost with that, I will hand it over to Jeff, who can tell us actually how not to get lost with single cell.
Thanks, Marta. I feel I'm going to tell everyone how to get lost. All right. So my name is Jeff. I'm a Science and Technology Advisor with 10x Genomics, and it's my role within the fellowship program to relay all the technical aspects of our single cell products, along with having one-on-one project discussions with all the successful applicants. So as Marta said, my goal is to make sure we're not getting lost. Before I start today, I just want to quickly say thank you so much for joining us here, and thank you for thinking about applying for the Millennium Science Fellowship Program. You're likely going to hear the same message repeated throughout the launch event, but it really has been such a valuable program for so many first-time single-cell users. Single cell can definitely feel very daunting, but by going through the fellowship program, you have, this additional layer of both financial support and expertise that means you'll come out at the other side an expert in single cell. Rather than just introduce myself, I want to showcase the entire local team that you have at your disposal for this fellowship program. There's your sales team up the top here. They are here to help you get your project up and running in the most economical way possible. There's, of course, myself. I'm here to relay all the technical information about our products and to have that project discussion or discussions if we need them. And then finally, we have our amazing support team who are here when it comes to all things on the wet lab. That's, trainings, right through, if needed, troubleshooting efforts. The take home really from this slide is that every person you see here shares the same goal, which is to make your single cell experiment a success. whichever way we can. And to show you just how dedicated we are as a team, here's our field applications team. They're ready to rock and roll at any stage during your single cell fellowship. We can deploy them straight into your lab. All right, I want to kick things off today with this slide that sets the stage really to the philosophy behind why we build the products we do at 10x Genomics. To make any progress in unraveling the complexities of biology, you're going to need the right tools at your disposal. You need technologies to provide insights at the fundamental resolution of biology, which is the resolution of a single cell. It's this need that underpins the direction we take when building our products at 10x Genomics. And you can see here that we've built 3 platforms that each allow you to look at single cell biology with a unique perspective. We've got Chromium, Visium, and Xenium platforms, which are shown right here. Focus of today's presentation, and of course the fellowship as a whole, is our Chromium single cell platform. So let's explore this in more detail. And I'm going to begin with perhaps an obvious question, and to be honest, one that I assume you already kind of know the answer to if you're applying for the fellowship, and that's going to be, why do single cell sequencing in the first place? These next few images, they're not super in depth, but I think they provide a really powerful demonstration as to why you'd want to do single cell sequencing. And here we have a blood sample, same blood that's pumping through you and I right now. Of course, blood is made up of a large mixture of different cell types and cell states. So when doing the more traditional bulk transcriptomic measurements, the whole sample is processed as a single measurement. And that's what you see here, just a single dot. You can tell the expression profile of the entire transcriptome, but that represents an average signal across all the cells in the sample. But here's what happens when you perform a single cell sequencing experiment on the same sample. Instead of a single averaged whole transcriptome, like we get out of our bulk sequencing, now you're able to measure whole transcriptome from each and every individual cell. This enables a significantly more deeper profiling of the sample, and you can now do things like identification of distinct cell types, states, and lineages, which of course in turn uncovers the cellular mechanisms underlying the health and disease state of your sample that you're studying. Hopefully it's pretty clear just from these images on the slide as to why you'd want to do single cell sequencing in the first place. Blood example, it's an easy way to conceptualize the power of single cell sequencing over using a bulk approach. But expanding on this, over the past decade, single cell has truly revolutionized our understanding of biology across such a broad spectrum of disciplines. And by now, I'm sure that you've all read at least one paper within your respective disciplines, which has used single cell sequencing to some capacity. Typically, I like to go through these categories and highlight some amazing literature examples that have used single cell sequencing to derive some sort of deep biological insight. I'm not going to do that for today. Instead, I'm just going to flick through these and highlight that there is an ever-expanding list of publications that show some groundbreaking, and in this case, pretty large-scale single-cell studies, and that they have all led to brand new understandings of biology in ways that bulk sequencing just can't deliver. So by now, it's very clear as to why you want to do single-cell sequencing. But what are the key pieces of information that you need to know before undertaking your actual experiment? The first and most critical thing to know about any single cell experiment is that sample preparation is key. And I cannot stress this enough, just how important sample preparation is. It makes or breaks your single cell experiment. Kat, who leads our field application team, is going to talk about sample preparation in just a moment, but I will say that your sample preparation journey really begins with 2 simple questions, but the answer to these questions will have deep implications as to how you go about dealing with the actual samples you've got. First question, what sample type are you actually working with? Is it PBMCs or cell lines, which are pretty friendly to deal with? They're already cells in suspension quite often. Or is it going to be a tissue sample that comes from a surgery at a random time of the day and arrives to you in some sort of random condition? That's a much more challenging sample to deal with. Next question is, what are your research goals from this single cell experiment? So if you know your sample and you know your biological questions that you're going to ask from that sample, then we can begin to map out your sample preparation journey. And through this journey, there are a lot of considerations. I've listed a few here, but I could fill this entire slide with things to consider. Each one of these will really dictate what needs to be done for sample prep. And again, Kat's going to discuss some of these considerations in more detail in her section, so I'm not going to take a deep dive into this. Instead, I want to really have you leave with these 2 points. Number 1, sample preparation is key. And number 2, practice sample preparation whenever it's possible to do so. Don't leave it to the day of a single cell experiment to be the first time you're actually going to try sample preparation. Beyond the golden rule at the top here, which again is sample preparation is key, there are some other things to know before starting your experiment. To be honest, we will cover this when we have our one-on-one technical consultation, which is part of the fellowship process. So I'll save some time and not go into great detail of each of these. I will linger on this last point, which is that our support website has a number of very valuable resources that you might find useful before starting your experiment. I've going to quickly walk through some things on our support site just to highlight what you can find there and how this will help during the experimental planning phases and, to be honest, beyond. So this is what our support site looks like. You can see that we have our support page listed out for each one of our individual products. So, we'll click on the support page for the single cell 3' assay. And this contains a bunch of resources from protocols, user guides, and free to use analysis tools. And you can see that all that information is laid out on the left. There's a lot of content on here. All of it's extremely useful. The content is laid out in the same flow as your experiment. So you start from the top of that list with experimental planning and sample prep, and then you work your way down. There's no need to be familiar with all this content at the start of your single cell experiment, but as things unfold, this page is going to become very useful. One of the great things we have on most of the product support pages is how-to videos. There's various levels of wet lab support associated with the fellowship that we can offer in order to help your experiment be a success. But at a bare minimum, if you're not sure about the single cell workflow or just want to build some confidence in executing the experiment, then these videos are amazing. They're just a few minute snippets of key steps in the workflow. Personally, I'm a visual learner, so for me, I think that's a really great way to learn this, just to watch someone go through step by step. Our support page isn't just documents and videos relating to our products. We also host knowledge-based articles, and we get asked the same questions all the time. So what we've done is curate the answers to these common questions within these articles. And these are a great way, if you've got questions, but you don't feel like you want to reach out to us personally, you can quickly go up to the website and see if you can find an answer to them. If we click the Q&A button, it takes us to this page. The articles are laid out according to what product they're associated with. You can also just type into the search bar here keywords to help find the right content. So if we click on this article and go into the 3' gene expression assays as an example, then all the articles are laid out according to which part of the workflow you have questions about. And again, these are super detailed and a great way to get some answers without having to write an email to us or find the time to speak to us. All right, so we know why single cell is important and what we need to do before starting any single cell experiments. And just because I haven't drilled it home enough yet, sample preparation is key. So before you start your single cell experiment, you want to know more about your samples, what biological questions you're asking from these samples, and ideally have practice and sample prep. But now it's time to learn what single cell assays are available, which really means what could I measure from a single cell? Over a decade of single cell product development has now been condensed down into the product portfolio that we have at this present day, which is of course what you see here. There are 3 product families in the Chromium single cell portfolio. They're the universal assays, the flex assays, and the epi assays. So we'll learn more about what these assays can actually do. We'll start with our universal 3' gene expression assay, or you might otherwise know this is just the 3' assay. This uses a reverse transcription-based biochemistry to directly capture and sequence the 3' end of polyadenylated transcripts. This means the technology is species agnostic, and beyond simple gene counting, you can also extract transcript level information like isoforms or express SNPs. It also means that your samples will need to be prepared fresh on the day of your experiment, which can have logistical implications that you certainly need to plan for. Anyway, using this kit, you can obviously measure whole transcriptome gene expression, but you can also extend the menu of analytes to include cell surface proteins. Now on the right, we have our universal 5' gene expression assay, which captures the 5' end of polyadenylated transcripts. And again, you might just know this is the 5' assay. This assay is also reverse transcription based, and you can also measure whole transcriptome gene expression and also capture cell surface protein information. But because we're now assaying the 5' end of the transcript, it's also possible to extract full-length paired VDJ information from T cell and B cell surface receptors, or TCRs and BCRs, if you will. It's also possible then to extend the menu of analytes to include direct capture of guide RNA molecules if you're doing any CRISPR screens. Really, to put this more simply, if you're interested in whole transcriptome gene expression, the majority of people will use the 3' assay on the left. But if you are also interested in that immune repertoire or CRISPR screens alongside whole transcriptome gene expression, then you would choose the 5' assay on the right. Besides the differences in these analytes that you can measure, obviously listed here on the slide, the workflow and specifications are basically identical between these 2 assays. Next, we have the gene expression flex assay. Yet again, the core measurement of this assay is obtaining whole transcriptome gene expression, and you can also extend the menu of analytes to include cell surface proteins. You can actually do intracellular proteins with this assay, and you can also do CRISPR guides. So this begs the question, why do we have a 3', a 5', and a flex assay, all capable of measuring whole transcriptome gene expression? The key selling point for the flex assay is that samples are fixed as part of the sample preparation process. So now that actually decouples the sample preparation process from running the single cell workflow. It means you can build up a bank of samples, which you can then batch process all at once. And this is huge for a number of researchers. Again, like I said before, sample preparation is the most critical aspect of any single cell experiment. But with the Flex assay, you can that now be done with a more standardized workflow. It means no compromising cell quality by rushing to get onto the instrument and use the single cell workflow. And as you see here, you can take either whole tissue slices or your suspensions of single cells or single nuclei. And rather than having to prepare and run them straight away, you simply fix the samples using 4% PFA and store them for later use. Another exciting aspect of the Flex assay is that it also works well on FFPE samples. So there's no need to modify the actual single cell workflow when using FFPE samples, just the upfront sample preparation, which allows you to go from this FFPE block to a suspension of single cells. And we, of course, have protocols to walk you through how to do this. Obviously, significant for those of you who have FFPE samples already collected, or if this is the primary sample format that you're used to working with. So if the Flex offers all the sample preparation benefits that I've banged on about, especially how important sample preparation is, then why not just use Flex on any sample? The biggest trade-off for the Flex assay is that it uses a probe-based biochemistry. Remember the single cell 3' and 5' assays, they use reverse transcription. So we're directly capturing and sequencing transcripts themselves. And this makes those assays species agnostic. That means you can access transcript level information. But for Flex, since the samples are fixed, this degrades and cross-links RNA molecules, which means we can't measure them directly anymore. So instead, we have to hybridize probes to transcripts and then barcode and sequence these probes. So basically, if you sequence probe A, then we infer that transcript A must have been present. But we only have probe sets for human and mouse, and those probes only enable transcript counting. So we're not accessing any transcript level information with these probes. So if you have human or mouse samples and just want whole transcriptome gene expression, then this assay is going to be a game changer for you. Finally, we have our EPI suite of assays. This comprises of our single cell attack and multi-omic assays. With single cell attack, you're measuring genome-wide open chromatin regions, so moving away from RNA to DNA. Then with multi-omic, you're actually measuring genome-wide open chromatin regions alongside whole transcriptome gene expression from the exact same single cell. I'm not going to be covering any more about these assays beyond what's on this slide, but I will say that multi-omic is quite popular since obviously you're directly linking gene expression and epigenomic profiles from the same cell. However, I would say that it comes with some of the most sample prep considerations and demands. So if you're thinking about using multi-omic, then we will most certainly need to discuss how to optimize and practice the sample preparation for that assay. Quick recap at this stage. We know single cells are important. We know what information we should know before getting started on your single cell experiment and what single cell assays are available to you. But now let's address which one of those single cell assays is going to be the right choice for your study. We, of course, are going to meet and discuss your project in great detail in order to derive this answer, but I also want to step through the following table, which has some considerations which can also help define which assay to choose from. So firstly, the species you're studying. If it's human or mouse, then great. You can use any assay at this stage. But if you're outside of human and mouse, or even using, say, xenografts, combination of human and mouse, then you're going to need to use either the single cell 3' or the single cell 5' assay. And again, that's because those assays employ reverse transcription-based biochemistry, which captures polyadenylated transcripts directly, and as such, are species agnostic. Next very important consideration is what is your sample type? To be honest, all the assays will work with just about any sample. It's more of a question around, can you freshly prepare that sample? And if so, then any assay is applicable. But if you can't, then you're going to need to use the Flex assay or think about some sort of freezing of your samples. Flex assay will also work with FFPE tissue, as I just described, but also PFA fixed tissue and fixed whole blood, whereas the other assays won't. What you want to actually measure plays another critical role in assay selection. So all assays are capable of measuring whole transcriptome gene expression. But if you're interested in analyte classes beyond this, then each assay has its own considerations for how to obtain those measurements. We just went through this, but as an example, if you're interested in immune repertoire profiling, so measuring those TCRs and BCRs, then you must use the 5' assay. But if you're after cell surface proteins, say, then all assays can actually measure this, but there are specific antibody assay combinations that need to be considered. Next is instrument compatibility, which really means instrument access. There are 3 versions of the Chromium single cell instrument, each capable of running different assays. I'm not going to go into deep detail here about what instrument can be run with which assay, but I do want you to know that through the fellowship program, there are options to get you the instrument access that's needed. And actually, Marta touched on this at the start, but it's one of the key features of the fellowship program that's helped many people in the past. Final assay consideration is throughput. To be honest, I think another way of looking at this is rather than static throughput, I really mean what different multiplexing options exist. For those who are unfamiliar with multiplexing, instead of running one sample per reaction, you combine multiple samples together and you run them together in one reaction and then use informatics to determine which cell belonged to what sample. Everyone wants to multiplex. I don't blame them at all. Obviously saves a whole bunch of money, but it can come with some risks and will complicate your sample prep. So take-home message here is there are different multiplexing approaches associated with each assay, and we can obviously discuss their merits during our one-on-one conversations. So walking through that table was all well and good, but I wanted to distill it down to some clear messaging around which assay to choose. So if you have human or mouse samples, just interested in whole transcriptome gene expression, then I would use the Flex kit. Also, same goes if you've got FFPE samples. But if you have non-human or non-mouse, or you really want transcript level information alongside that whole transcriptome gene expression, then single cell 3' is the way to go. Also, this is going to be the right assay to use if you have really low cell numbers, say something less than 30,000 cells in total. And finally, if you're interested in the immune repertoire being those paired TCR and BCR sequences, then 5' assay is right for you. And also, if you're doing CRISPR screens and want to read out of that guide RNA molecule in addition to whole transcriptome gene expression, then this would definitely be the assay I recommend too. The reality is choosing the right assay is dictated by your sample type, sample preparation strategy, desired biological measurements, and multiplexing. All these things can mean compromises and can actually make the choice, quite difficult. But again, never fear, we'll have that initial project consultation, and a key focus of that meeting is going to be helping choose what is the best assay for your project. So the final aspect I want to touch on in the last few minutes is data analysis for single cell sequencing. It might be at the back of your mind right now, especially since you haven't even run the experiment yet. But trust me, it's kind of like sample preparation. The more you can plan and practice your analysis, the better equipped you will be for handling your actual single cell data. So these few slides just provide a quick overview about analysis and the software that's required and the software that's provided by 10x. I guess the analysis journey for single cell data looks something like this. You start with raw sequencing data over on the left, and that's processed into files that can be biologically interpreted. Then the data is assessed for its quality, undergoes some forms of quality control and refinement, and next is the actual biological interpretation, which typically involves a cyclical process of annotating cells and finding differentially expressed genes. Beyond this, there are field-specific analysis that people employ, like trajectory analysis or pathway analysis. I would say that this analysis journey used to be very daunting for many researchers, but as the field of single cell has progressed, so have the analysis tools. I'd still highly advocate that you have some form of bioinformatics input into both your experimental design and data analysis, but these days, much of your analysis can actually be done without having a deep knowledge of informatics or coding. So you can progress through this analysis journey even just from your laptop with minimal coding information. So the last slide outlines the journey. Now this slide highlights the software tools that you need along that journey. And firstly, there's Cell Ranger that comes from us, 10x Genomics. This is a collection of analysis pipelines for taking that raw sequencing data and processing it into files for biological interpretation. Once you have that output from Cell Ranger, then you can use Loop Browser to view and analyze data. Again, Loop is another free software from 10x Genomics. It was designed to be a point-and-click style GUI for data analysis where you don't need to have deep informatics skills. So, the run's finished, very excited about my data. Let's quickly load it on the laptop. Right in front of me, here's the UMAP plot, and I can start interacting with that and actually doing some pretty heavy analysis in that tool. Beyond Loop, there are a huge number of community-developed tools, which are typically run in the command line using either R or Python. Although they're not officially supported by us, many of these tools are very useful to perform a variety of advanced analysis. So if we go back to our analysis journey, here is what it would look like when you overlay each one of those software packages. Again, you start with Cell Ranger to process your raw sequencing data. Then you immediately use Loop Browser to view the outcomes from Cell Ranger in a very simple point-and-click interface. Likewise, files from the Cell Ranger can then be also loaded to third-party software for further analysis. And we even have a small program called Loop R. So this is another 10x program, and this allows your informatician to do some analysis in the third-party tools like Seurat. Then you can use Loop R to make a file out of those third-party tools that can be opened by Loop Browser so that you can do more of the biological interpretation of this data without having to write code. You can just do it in point-and-click style on your computer. I want to make a quick note about community-developed tools, as they're obviously very powerful, very popular, something you're definitely going to use. However, since we don't write the code, then it's hard for us to support these tools. Also, many of these tools do things that Loop can't do, which is why they're very popular. But we wanted to help the single cell community with your analysis. And that's why we came up with these analysis guides. They're a collection of introductions, tutorials, and blogs for data analysis beyond software developed by 10x. So I've included links to the more popular articles here, and I'll share these slides, through Marta and then go out to everybody here today, so you can access this material. Along with the analysis guides, I want to give a shout out to our new completely freely available 10x Genomics cloud. What does this cloud actually do? Well, within the cloud, you can run the Cell Ranger or Space Ranger analysis pipelines to process that raw sequencing data, again, into the output files ready for biological interpretation. Before the cloud, you need to install these Ranger pipelines on a local Linux server to run them. Generally meant you needed to find and pay for informatics support just to process and store your data. So I would highly encourage you to make a free cloud account and start running those analysis pipelines using a simple web interface to get your data quickly. All right, wrap things up with this slide. Key pieces of software, again, a Cell Ranger, Loop, and community developed tools. For our free software packages, there are tutorials and lots of documentation on our website to enable their use. And for community developed tools, many of them, particularly the bigger packages, also have great tutorials and instructions, and you can always head to our analysis guide website to help use them. With that, I'll conclude on this slide. Before I hand it over to Kat to talk about sample prep, I just want to say thank you once again for coming along today, and thank you for hopefully applying to the Millennium Science Single Cell Fellowship. I can't wait to meet each and every one of you in our one-on-one project consultations, and more importantly, honestly can't wait to be reading a bunch of single cell papers in a year's time that have all come from your successful experiments. So I'll hand it over to Kat.
Great. Thanks, Jeff, and hi, everyone. So my name is Catherine King. I am, as Jeff and Marta both mentioned, I look after the field application scientist team here across all of Australia and New Zealand. And our role really is to help support you, once you've decided your assay, you're getting the kit in, and we're there to kind of help you use that kit, right? We have chats with you about your sample preparation. You've already heard, quite a bit about sample preparation and how important that is. So we can really help work with you on that, as well as, talk you through the specifics of the assay and make sure you've got all the relevant user guides and documentation, everything you need. So what is sample preparation? So it's really what you bring to the experiment. So the reason I have this slide here is the key usually, Jeff's introduced quite a few assays here. We've now got ways of, inputs of FFP samples and fixed tissue, but generally our input into a single cell assay is a single cell or nuclei suspension, no matter what you're actually bringing in. Even if you're working with FFP samples, it'll end up as a nuclei suspension. And we're going to load that onto our Chromium instrument. So what you really need to do is have a really nice high quality single cell or nuclei suspension at the end of your sample preparation workflow. And how you get there is going to be so dependent on what sample you're working with. So if you need to kind of ask yourself, what does this involve? So what organism are the samples from? Are these, patient samples from humans? Are they going to be coming from a hospital that's nowhere near where you're located, and somehow have to get to you in as high a quality as possible so you can dissociate them into single cells? Or is it a cell culture that you're working with in your own lab? Is there a specific tissue or cell type you're interested in? So this will really define what the sample prep will look like. If it's, blood, the cell's already in suspension, quite nice workflows already developed to isolate cell types of interest from blood, as opposed to, maybe taking some brain tissue and having to dissociate into the single cell suspension. So this is really going to make a difference to how your cell preparation looks. And then, do you already have a workflow for getting a really nice high quality single cell or nuclei suspension, or is this something you're going to have to develop and get working in your hands before you go into a single cell assay? And these are key things to ask yourself now, because this is something you can do as you're applying for the fellowship. You can start thinking about these so you're well and truly on your way to be ready to run a single cell experiment. The other thing to consider is just what you want to analyze. So Jeff's introduced you to the assays and, at a very high level, what we, offer from 10x Genomics, but really your sample prep and what you have to, whether it's a single cell or single nuclei suspension, it's going to define what kind of analytes you can look at. So you may need to think about your sample preparation if there's specific things you're looking to gain from, and information you're trying to gain from your single cell experiment. So if you're looking at gene expression, and then you also want to look at protein expression, so you want cell surface protein expression, or you want to look at VDJ sequences, then you need to be working with whole cells. On the other hand, if you really want to look at and use some of our epigenomic products like our ATAC, our multiome assay, you were going to have to prepare a single nuclei suspension for that assay. So we say sample quality is critical. You've heard it now many times already in this presentation. But what does that really mean? So age-old saying, rubbish in, rubbish out. It's kind of true though. So if your cells are nice, high quality, intact cells, the membrane's intact, so all the RNA transcripts are inside the cell and not kind of leaking out. They're, not in a state of biological decomposition, so they're, shutting down transcription. You're going to get really high, nice, single cell data from the cells and low background, giving you nice single cell data. If you have compromised cells, whether that's physically compromised or biologically compromised, then that membrane's breaking down, transcripts are being released into the background, they're shutting down transcription, and now you're going to have high background, you're going to have poor single cell data. And so this is the reason we talk about so much around sample preparation. So what actually makes a good quality sample? We've got a measure of cell viability here. If you were working with cells, saying that 70% pretty like quite a minimum cutoff. We really want higher than that, as high as possible. High viability kind of equates to nice high quality data. We want our cell suspension to be single cell, free of aggregates and clumps, free of subcellular debris and background RNA and DNA. Remember, we're working with a fluidic system, so it has to be these single cell nucleases to flow through nicely. We want them in a minimal state of biological decomposition. So this means not only they're, nice single cells and currently viable, but they're also healthy because, that's where we're going to have our transcription as we expect in that biological sample, as opposed to already cells membranes compromised, leaking into the background, getting high background. And then physically intact. So we want to prepare them quickly, minimal time before moving from having our sample ready to loading on our instrument so that they're nice, physically intact, again, for that same reason, low background, high signal from our cells. So what does a sample preparation workflow kind of look like? And really, there's multiple stages of a sample prep workflow. And this is an idea of what it could look like, is collection of your sample, preserving the sample. Some of these may not, be relevant for your particular sample. Again, if you're working with a cell line that's in your lab, you're not going to have to think about transporting it to your lab. But these are all possible things you might need to include in your own sample preparation workflow. And what we do have on our website is demonstrated protocols, the Q&As that Jeff mentioned, that kind of address each of these points and gives you some ideas and helps you build your own appropriate sample preparation workflow. One of the key places to go on our website and to get started is to think about sample preparation and looking at that sample prep resources tab on the 10x Genomics website that Jeff already kind of showed you where it is. That's a great place. We have lots of protocols there, from really general guidance to very specific protocols for certain cell types. So great place to start. And also, if you're looking for one guide to have a read of now to get yourself really aware of what you need to do for sample preparation for single cell analysis, then my suggestion would be to look at our cell preparation guide. This is recently updated. It has lots of nice content in here, everything from buffers you can have your cells in, things to avoid in your sample preparation, how to do flow cytometry to clean up your samples if you've got lots of debris or dead cells, all these things. So this is all in this cell preparation guide and a great start to really get familiar with what you might need to consider for your own sample prep. That's all I had. I only had a really short time to give you a quick spin around sample prep. But what I will say is that if you are part of the fellowship, just like you'll have a chat with Jeff about your experimental planning, once you're getting started, you'll also have a meeting with, and have multiple meetings likely, with members of my team. And we will talk you through your specific sample preparation workflow, make sure, give any advice we can related to that, help you talk about these pilots and see how that's going, and then also talk you through the actual 10x workflow and what you need to consider from that side of things. So thank you for your time, and I will stop sharing and hand back to Marta.
Thanks, Kat. Thanks, Jeff. So we can now move to our next step in our today's meeting, which is a quick panel discussion with our previous fellows, Jennifer Boer and Dmitry Shek. So guys, you can unmute yourselves because I might ask you some difficult questions. Okay, so Jennifer, Dmitry, what was your favorite part of the fellowship? You can be honest. You should be honest.
I might start there. So Marta, yes, one of my favorite parts of the fellowship was indeed community. Alex mentioned this in the beginning, and I think community is not just a matter of support, which is really good and well to have, but I also found that in community is also support and where the difficulties or where we bump into difficulties doing single cell. And I find it also, I found it incredibly valuable to learn from other people's mistakes, because that kind of avoids you having to to find out yourself. So the fact that there's community and you can exchange ideas and and realize that some of the problems that you may be bumping into, other people have it as well and have resolved it differently, because ideally your cells are perfect, but for us at least sometimes unfortunately that is not, and so you have to find different ways to navigate and still go ahead with your experiment. So community for me, Marta.
Dmitry?
Well, yeah, I also agree with Jennifer. The program has been an incredible experience, and it provides not only access to this cutting edge technology, but also this invaluable mentorship from Jeff, Kat, Jerry, and also this strong collaborative network. From my side, for example, the support that I received, it significantly enhanced my research and my PhD progression. It not just allowed me to apply these innovative single cell and spatial transcriptomics approaches, which, by the way, we did it for the first time at Westmead Institute. But beyond those technical aspects, this fellowship fostered the community of like-minded researchers. And I remember this dinner which Alex organized last year in Sydney, and where we were able to meet and to, enable those meaningful discussions and collaborations, which still continue to shape my work today. So for me, it was a transformative experience, and I truly encourage everyone, all new applicants, to take this opportunity, because it will definitely empower your research, and it will open the doors to such an exciting possibilities, and it will definitely elaborate your research or your PhD, or whether you're a postdoc. It will definitely enhance your research and move you to the next level.
Thank you. I'm blushing on behalf of Millennium Science and 10x Genomics. Jennifer, Dmitry, do you have a favorite 10x Genomics essay? Or do you love them all? I understand.
Yes, obviously. Well, they're all incredibly exciting, right? I mean, in our lab, we do a lot of vaccine study that is based on immune cells. So the single cell itself gives us so much more insight and exciting stuff to find out about what specific cells that we do in the lab. And so for me, it's, aside from the 5' assay, it's the VDJ library for T cells. So the ability to then go in to say, okay, right, I have my cDNA library from all the cells, and now I want to hone in on the T cells, and I can look into what the diversity looks like. And sometimes if your cells are not too rare and you can just, you don't have to sort for them up front, you can sometimes also use what remains from your 5' experiment, and then actually apply that for a VDJ library, which I found also incredibly useful, because then when you find exciting stuff, you can go and it's all right, well, let's have a closer look. So that's one of my favorite parts. Yeah.
Dmitry, do you have your favorite essay? Or is it the same one?
Yes, I partially agree, but among so many remarkable technology that 10x Genomics can provide, I think for us, Visium, and maybe if we have an opportunity to try Xenium in future, they are particularly transformative for my research, because we are working on the immunotherapy-related colitis, the specific condition which some cancer patients develop after being treated with this transformative immunotherapy. And for us, in particular, Visium has provided some unparalleled spatial insights, and it allowed us to map gene expression with the tissue architecture and to potentially discover some new biomarkers which can predict the development of immunotherapy-related toxicity in those patients, and to the ability to identify those tissue-specific biomarkers of cancer immunotherapy. This is definitely going to shape the clinical oncology within the next 10 to 20 years. So yeah, I probably would say that all technologies are superb, but for us, Visium and potentially in future Xenium will be something what definitely makes change for our research.
Yes, we are also waiting. Jennifer will jump on spatial too, but
we've got special plans for spatial.
Spatial is very spatial, but also single cell is very spatial. Special. Well, we are running a bit out of time. Of course, we will go through all the questions and answers. If in our audience, if you have questions for Jennifer and Dmitry, while I will be walking through all the questions that are there so far, you can also put questions for them. Just finally, I wanted to just also say that Jennifer and Dmitry, like a few years ago, they were just like you are right now, didn't have experience with 10x Genomics, and now they are really successful in running the experiments themselves.
Yeah, so I would like to add to that, Marta, because also, like, we talked a bit about community from, and for other hardships and other people, but it's also community within the 10x team, and that I found really soothing, because starting a complex new experiment can be very daunting, but knowing that you can describe and plan and have someone at your side as well in the lab to go through that, I found that incredibly helpful.
Yeah, I think I remember this moment when Jeff was telling me that every single day he had called from Jennifer about something. Okay, well, we are very serious. Jumping into questions. Question number 1, I have experience with Xenium, but not single cell sequencing. Am I eligible for this fellowship? Yes, you are. This is single cell focused fellowship, so you can feel free to apply. How many fellowships are on offer? So we don't have really hard cutoff with the number of fellowships. We will never reject really a good, fantastic project. So you don't need to worry about like really limitations. Of course, we can't accept, everyone, but we do what we can and to expand the number of accepted fellows as much as we can. Next question. Do we need to have funding in place to perform single cell experiment to apply for the fellowship? Well, it is the fellowship, and I saw the question later, provides discount for the reagents. It's not free reagents, so having funding or having support from UPI with funding, it will be really essential. However, we can also support fellows who are at the stage of writing grant applications. So usually the fellowship is given for a year. So this is when we expect you to conduct your experiments but with several cases, we know how the science works. Sometimes it does, sometimes it doesn't. So there is always possibility to extend the fellowship for a longer time. Can graduate student and ECR put in one application together? Yes, we had that in the past, so it can be a joint fellowship. So this is accepted and possible. Are PhD candidates third year eligible for it? Yes, they are. All the early career researchers basically are encouraged to apply. The discount, what discount do successful applicants get? This is 20% discount on the reagents, and which is not actually mentioned in the value. There's lots of value in the fellowship, which is not really costed in terms of the reagent. The time that actually Jeff and Kat and all the meetings, that's really actually can be costed in a dollar value, but it's like additional thing that is offered with this fellowship. Okay, same question. So PhD and MCR in application, yeah, that's absolutely fine. Are you looking for innovation or feasibility in the application? I would say both, but I think in case of 10x, as we heard Jeff and Kat, feasibility is really important. We don't want you to commit yourself and get yourself into experiment which has high risk of really not working, especially knowing how much effort, time, and costs it can entail on your end. So definitely feasibility is something we are closely looking at. Will 10x Genomics need to share the patent of the project if fellowship accepted? I understand it is a question whether you do your research using 10x Genomics reagents with the fellowship, and whether if you patent something, whether 10x share the patent with you. No, this is completely unrelated. The patent belongs to you. Publication is yours. However, putting little acknowledgements with your publication to Millennium Science and 10x would be nice. And we have few more questions. Next one. Oh, now we have some technical questions. So Jeff, Kat, be ready. Will intra-seq be possible with 5' assay in the future? And why is fixing not amenable for the 5', 3' assay? Is RNA integrity worse with fixing?
Yeah, good question. So intra-seq, I'm not intimately familiar with. I'll go read that publication and figure out if it can be adapted within the workflow. Quite a lot of we call them customer-derived applications. Actually, hacking of the kit will work with our kits. People do weird and wonderful things, so hopefully that's going to be the case. When it comes to fixation, fixation does cross-link RNA, but not necessarily degrade RNA. So when for the flex assay, when we fix samples, we don't see a reduction in the performance of that assay in terms of sensitivity. In fact, until recently, it was one of our highest sensitivity assays available. But it does mean that you can't use reverse transcription to access those molecules anymore. So that's why we have to use a probe-based workflow, but that probe-based workflow for fixed samples is as sensitive as if you were going to use them fresh. We do also have sample fixation options available for the 3' and the 5' assays. They're a reversible fixation. So it's not using a cross-linking agent, it's using something else. We do so in that context, if we were to apply this reversible fixation reagent to the 3' and the 5' assay setting, that there is a slight reduction in performance. But I wouldn't say that it's anything of significance.
Okay, and yeah, did you answer why is fixing not compatible with 5', 3'?
Oh yeah, I think I did, but I'll just rehash it.
Yeah.
I probably didn't. So once you fix the cells again, the RNA molecules become cross-linked. We can't use the reverse transcription-based biochemistry of the 3' and the 5' assay on cross-linked RNA molecules. The enzymes just don't process through their actual molecules and they fall off, and we just don't capture their signal. So instead, we use the probe-based biochemistry. So if we're fixing, we've got to use the flex assay with probe-based biochemistry. If you're not fixing, then we can use the 3' and the 5' assay.
Yeah, and then there is also an option for some temporary fixation, which can be discussed during the project discussions. Next question, when the recording file will be sent? Well, it will take a few days for the process the recording and upload it, so it will be sent, the link will be sent to everyone who registered for today's webinar. Technical question, is there species agnostic VDJ sequencing?
In theory, it's possible. So our kits are designed for either human or mouse, TCR and BCR, but we do have guidance on how to design your own primers to extract that from non-human and mouse species. So Jennifer touched on it earlier. What we actually do is we create a library of molecules that contains all the molecules from a single cell. And then from that, we're going to enrich out the VDJ sequences. And we do that using PCR. So we've got guidance on how to design, well, you design the PCR primers, but we've got guidance on how to use your own custom PCR primers to extract TCRs and BCRs from non-human and non-mouse. Have seen it successfully implemented in pig, sheep, and there are certainly customer publications out there.
What are the considerations with an experiment consisting of primary neutrophil and bacterial co-infection, which must be halted at specific time, especially in regards to cell viability?
Flex. Flex is a great example of how to use this. Sorry, Kat, I cut you off.
No, that's all right. I mean, I was going to say that. I mean, if this question, I guess we'd have to delve into this a little bit more and the considerations around it from a, from your work perspective and how you get these samples and how many samples you have and batching. But ultimately, Flex, as Jeff alluded to already, allows you to fix those samples so they are locked in place. So although you think, oh, fixing samples, all this talk about viability, how does it matter? Well, it matters at the time of fixation. So it's all the same principles, but instead of the time of putting into our Chromium instrument, it's the time at which you fix them. But if you have a population of cells, you need to capture them at a very specific moment, but then you're going to have multiple samples you want to batch into the same experiment. Flex allows you to do that, and there's a huge benefit of it. The other, otherwise, if you're not able to do that, you want to work with fresh cells, it would be being at, you literally need to be with those cells at that point in time, capture them at the point of, at this specific time you need to halt that experiment and run into the single cell assay. If the question's in regards to viability because of the co-infection, I guess that's part of the biology and the question you're trying to ask. And so that would need some consideration to make sure you're going to get, the data you need out of it.
Thanks, Kat. For the abstract, as part of the application process, do we need to outline sample sizes of the human biospecimens we wish to analyze, or shall we keep the abstract high level? And then there is a next question related to that. Is there a specific format we need to follow for the abstract? So abstract is just pretty short, just outline what you want to do, why you want to do it, why is it important, of course, and what, very general overview of your project. There will be, during the application process, there will be several questions, including how many samples do you have? Are they like fixed, fresh? What species? So these specific questions will be in the application form you will be filling. Going to next questions, we have quite a lot. How exciting. How often is one-on-one consultation? Where is it? So we usually start online. We can do it in person depending on how the team is traveling. How often it's really tailored to needs of yourself, of your research project. How complicated it is everything sorted? So we are always there, basically. You can ask for another meeting if it's necessary. And we are there to basically almost hold your hand when you do the experiment. I have analyzed single cell RNA-seq data in the past, but have never been involved in the experiment part. Am I eligible for this fellowship? Yes, you are. You are actually really positioned well because you already know how the data more or less will look like. So, and you have the skill to analyze it. So that's actually fantastic. Just for more clarification, if the fellowship is accepted, then are we going to send the samples how and where? So there are options that you should be doing your sample preparation, and you can run the samples yourself if you have access to instrument, or potentially use a facility, a core facility that can run the samples for you. We always try to keep in touch with the fellows to not get suddenly you disappeared from and stop responding to our emails. This is really heartbreaking. But we are not running the samples for you. So we are just providing the guidance, the reagents, and help you with the logistics. So it's based like project by project. It's we just discuss it with you. Do we need to talk about impact of the fellowship in the abstract? I think we all know that all research is really important, and I know you need to put the impact statement in your standard applications for funding. You don't need to overly really justify why your project is more important than someone else's project. We understand that. More is the visibility, importance, what do you want to achieve with that fellowship, with this research project. Track record is not that important, someone asked, but there will be questions about your experience with RNA isolation, with cell culture. So that is more here important than amount of publications and attending conferences. Could you do antibodies and GD, VDJ, seq? What's GD? I don't know what's GD.
Assume gamma delta.
Ah, maybe.
Short answer is yes. Most certainly you can do cell surface protein and VDJ sequencing. The gamma delta is a little bit more complicated in that we don't sell kits to do gamma delta expression. However, it is possible to do within the constructs of our assay. So certainly we've got a lot of nice guidance on what the primer designs need to look like, where to order primers from, how to spike them in, and our analysis pipelines will accept gamma delta assemblies now as well.
Yeah, there was clarification about alpha, beta, and gamma delta sequencing together. Thank you for clarification. I know Jeff knows things, and sometimes my knowledge in immunology is a bit rusty. Last set for questions, I think, for today. What if you haven't set up the experiments yet? How will you address this in the application? I have no idea what my sample size should be. I'm doing organoid work. So I guess you have a certain idea what process you are studying with your organoids, what information you would like to get. So it can be like, it's abstract is 250 words. It doesn't have to be detailed. You have organoids of specific model, organ, you want to learn something about it. You can basically put it pretty briefly. Why is it important? And keep the sample numbers to really low. It will not that much affect the application. You can also email me and I can try to help you out with more specifics about that. Okay, so patient lines, but not necessarily number of organoids. So the number of samples, as also Jeff mentioned before, there's many ways of multiplexing samples. So in the past, we had fellows that submitted application, they have like 50 samples. In the end, it was actually, let's say 4 or 8 really reactions because they were multiplexing samples together. So we don't look at it really strictly. If you say, I want to run 50 samples, you are really committed to run 50 samples.
Can I just add to Marta that also during our initial project discussions, if you even have no idea about how many sample numbers and cell number for that matter is appropriate for your experiment, we just sort of discuss this and help derive this together so that you kind of exit these conversations with a bit clearer idea about the whole experiment.
Yeah, I think you are at the stage you're thinking about it. So you are the expert in your field. You will know what you are looking for. Are there specifics that need to be included in the letter of recommendation from my supervisor? So this recommendation letter is basically to make sure that you actually discuss this application with your PI, that he's supporting you in terms of access to lab equipment, access to other funds or things additionally that might be required. So it's just very general that you have skills and to actually conduct the research. Okay, I think we managed to very much run over time, but everything is recorded. If you had to jump off, recording will be available. So
just Marta, there was one quick question as well that came up twice about instrumentation.
Yes. Yeah, sorry.
You don't have an instrument.
There was so many questions and I was just scrolling. Yes, so if you don't have instrument in your institute, there is a possibility you can ask to have a demo instrument placed for you and you can run it in your lab, or you can consider running through the core facility and send samples there. It will be discussed during basically onboarding meeting how you want to proceed with this. Some people are not feeling super confident to have instrument on their own lab bench, although I think it's an exciting opportunity to actually be able to do it. And it's also actually because the demo instrument is placed in the lab free of charge, this is additional cost in kind that is added to the fellowship, which is not only the discount on reagents. All right, I think there are no more questions. If you have any more questions to either Kat, Jeff, myself, or Jennifer, Dmitry, feel free to, you saw the email addresses, feel free to email us directly and we will definitely be getting back to you. Thank you, and thank you everyone from the panel. Thank you, Jennifer. Thank you, Dimitri, for joining, and have a great day.
Thank you.
Thank you.